Hello bachcho, how are you? So today I'll be teaching about immunity. Okay. What are the two types of immunity? One is active and one is passive. Here the antibodies are formed in our body when we get any type of natural infection. जब कोई एंटीजन या कोई मतलब जो कोई माइक्रो ऑर्गेनिज्म हमें इन्फेक्ट करता है तो हमारी इम्यून सिस्टम उसको प्रोसेस करता है और उसके अगेंस्ट एक इम्यून रिस्पॉन्स जनरलाइज्ड इम्यून रिस्पॉन्स प्रोवाइड करता है, तो एक एंटीबॉडीज प्रोड्यूस्ड होता है, तो उसको हम बोलते हैं कि एक्टिव इम्यूनिटी. नाउ इन पैसिव इम्यूनिटी the antibodies are passively injected. In active immunity, memory cells are formed. But in passive immunity, memory cells are not formed. So, as memory cells are formed, it is a long-lasting response. But here, memory cells are not formed, so it is short-lasting. It means it's gonna be persist till the injected antibodies remain in our body. Now, what is primary immune response and what is secondary immune response? Whenever our body sees antigen for the first time, it produces an immune response. So, the efficiency of this immune response will be a bit lower. But whenever body sees it for the second time, the intensity with which the immune response is formed, okay, it is this time it is much larger. Okay. So why this is happening? This is all because of memory cells. Okay. This is all because our body, once it sees any type of microorganism or once it sees any type of antigen, it keeps a memory of it, and next time when it sees, this time it recognizes it very early and produces the immune response with much more larger intensity. So suppose you are injecting first dose of antigen, means you are injecting an antigen for the first time. Initially there will be a lag phase, and then as the antibodies start to be produced, first they will rise, they will reach a peak. Okay, now it has become constant. Now you have injected second dose of antigen, the same antigen that you injected in the first dose, you have injected it for the second time. Okay, this is the second dose of antigen, second dose. This time first there is a decline phase. Why is this decline phase? Because in the primary immune response, the IgM antibodies are produced. Okay, IgM antibodies are produced, and the antigen that is injected will bind with this IgM. Okay, there will be antigen antibody complex, due to which initially the amount of antibodies will be decreased. Antibodies produce होने में भी कुछ टाइम लगता है, तो उसको बोलते हैं हम लैग फेज. तो यहाँ पे भी लैग फेस होता है, पर लैग फेस के आगे जो एंटीबॉडीज प्रोड्यूस हुआ था उसका तो एक लेवल होता है ना, उसका एक कांस्टेंट लेवल है, वो घट जाएगा बिकॉज की जो एंटीजन है वो एंटीबॉडी के साथ बाइंड करके एक कॉम्प्लेक्स फॉर्म कर रहा है. उसके लिए द एंटीजन्स दैट बेसिकली बाइंड डिक्रीजेस द रिमेनिंग नंबर ऑफ एंटीबॉडीज. ओके, सो दिस इज कॉल्ड द डिक्लाइन फेज. Now again there is a lag phase, again it will reach a peak. Okay, sorry तो interrupt हो गया. Okay, so this is the immune response. Okay, तो अपने यहाँ से secondary immune response. In the secondary immune response, the antibody that is produced is IgG. So from IgM to IgG, this phenomena is called class switching. I will be talking about it later. Okay. Now, 1 degree, second degree. What are the basic differences? It is mediated by IgM. Okay. It is mediated by IgG. Decline phase. You can see in the secondary immune response there is a decline phase due to the antigen binding with the previously or preformed antibodies. Okay. Due to which the remaining antibodies were decreasing. So there is a decline, no decline phase in the primary immune response. Okay, no decline phase, but in secondary immune response, as you can see, this is the secondary immune response. There is a decline phase. Decline phase is present. Okay, the peak is lower here. Peak is lower as intensity is lower. But peak here is higher. That means intensity is higher. That means here affinity of the antibodies for the antigen is low, means efficiency is low, but affinity is high here. Okay, as the body has seen the antigen previously, it can induce a stronger immune response. Okay, so these are the basic differences between another important difference is lag phase is shorter here. Lag phase shorter. You can see lag phase shorter, but lag phase, this one is a lag phase. Okay, this one is a lag phase. Okay, lag phase is longer. Okay, lag phase longer, that is almost 1 week. So these are the basic terms of immunity, and what is active immunity, what is passive immunity. So let's talk about MHCs. The questions mostly come from this segment. Okay, what is MHC? It is major histocompatibility complex. Okay, major histocompatibility complex. What are the types of MHC? There can be MHC1, there can be MHC2, there can be MHC3. Okay. Now what is the generalized structure? In MHC1 there are 3 alpha chains, okay, that are produced by chromosome number 6. Okay, and beta chain, beta 2 microglobulin that is produced by chromosome 15. Okay. Now HLA. Now there are different types of gene in MHC. We call it by HLA. Okay, human leukocytic antigen. Okay, it is the another name of MHC. It is another type of another process of representing MHC. Now in case of this MHC1, there can be HLA-A, HLA-B and HLA-C. Okay, these are the different types of MHCs. Now this MHC1 is present in all nucleated cells of our body. All nucleated cells of our body. Okay. Now MHC2. MHC2 is also having HLA-DP, DQ, DR. Okay. All these segments DP, DQ, DR, they synthesize 2 alpha chain, 2 beta chain. Okay. 2 alpha, 2 beta. 2 alpha, 2 beta. For H, there are 2 alpha chains and 2 beta chains, but in MHC1 there are 3 alpha chains, 1 beta chain. Okay. This is an important difference. Okay. And MHC3 is not that significant. It produces complement protein C2, C4, okay, heat shock protein, okay, tumor necrosis factor. Now MHC2 is present in professional APCs. Okay, now what is APC? APC is the antigen presenting cell. I will discuss about it. Professional APCs. What are the professional APCs of our body? That are basically B cells, macrophages, dendritic cells. It is the strongest APC in our body. APC, it is the strongest. Okay, it is the strongest APC. Now this MHC2, the MHC1, what is the basic difference between MHC1 and MHC2? The MHC1 is responsible for representing the intracellular pathogens, okay, the endogenous proteins, the intracellular pathogens, okay, the endogenous proteins means the protein is itself synthesized within the cell, okay, the protein is synthesized within the cell, endogenous protein. Synthesized within the cell. But MHC2, they are responsible for representing exogenous proteins. Means the proteins is basically produced outside the body, माने outside the cell, okay. लाइक अगर कोई बैक्टीरिया है बाहर, उसको एनगल्फ कर लिया, तो उसका जो प्रोटीन्स है उसको हम मतलब प्रोटियोलाइसिस करके जो प्रोटीन्स है उसको एक्सट्रैक्ट करके अगर हम रिप्रेजेंट कर देते हैं, दैट मीन्स इट इज एक्सोजेनस प्रोटीन बिकॉज इट इज नॉट सिंथेसाइज्ड विद इन द सेल, ओके, नॉट सिंथेसाइज्ड विद इन द सेल. Now different types of intracellular pathogens like viruses and tumor. Now here mostly the bacteria. Extracellular pathogens means bacteria, okay. Now this MHC1 represent the peptides or represent the antigenic peptides to cytotoxic T cells, okay, cytotoxic T cell, CD8, cytotoxic T cells, and the MHC2 represent the antigenic peptide to helper T cells, okay, THCD4. This is helper T cell. Now this all are the basic difference between MHC1 and MHC2. Now what is immunological synapse and how the process is occurring, okay, we'll discuss about it. Suppose this is a extracellular pathogen, okay, extracellular I am representing it with green color. Now there is an antigen presenting cell, okay. This extracellular pathogen, let it be bacteria, okay, bacteria, okay. Now this bacteria will enter inside the antigen presenting cell. Means antigen presenting cell will engulf the extracellular pathogen, okay. Now within the cell, suppose there is proteasomal machinery, okay, proteasomal machinery. This proteasomal machinery will break down the peptide into smaller pieces, okay, or this entire antigen into smaller pieces, okay, polypeptide into smaller pieces, and this smaller pieces will be represented on the MHC2. Now let me explain the difference between activation of Th1 cells and Th2 cells, okay. The helper T cells can be two types, one is Th1, one is Th2. The Th1 helper cells will come into action when there is intracellular pathogen, okay, intracellular pathogen, but there is exogenous proteins. Now you see here the MHC2 is responsible for representing the exogenous proteins in its cleft. Okay, and this represent to Th helper T cells. Okay, so you can understand if it is an intracellular pathogen and also there is exogenous protein, mean the protein is not synthesized within the cell. Okay, I am repeating it again and again. The endogenous proteins are synthesized within the cell, but exogenous protein are not synthesized within the cell. The microorganism is engulfed and the proteins are extracted from it through proteasomal machineries. Okay, तो उसको एक्सट्रैक्ट किया जाता है, उसको बनाया नहीं जाता है, इंडिजिनस प्रोटीन को बनाया जाता है. ओके, सेल के अंदर से मंगाया जाता है. तो TH1 for intracellular pathogen can be tuberculosis, mycobacterium, it can be leprosy. Okay, it can be Listeria monocytogenes. For tuberculosis and leprosy, the bacteria are intracellular, and for TH2, when TH2 come into function when there is extracellular pathogen. Okay, extracellular pathogen mean other bacteria. Okay, other bacteria mostly, other bacteria or other fungi. Okay, for fungi it is TH17 actually, so mostly TH17. So other bacteria. Okay. Now you can see, suppose this is extracellular pathogen. Okay, extracellular pathogen means it mostly resides outside the cell in the circulation, and the cell has trapped it from the circulation, and it is represented on the cleft of MHC2. Okay, cleft of MHC2. MHC1 will come into function only if it is endogenous protein, and it can be virus and tumor. Okay, so for rest of all that it is MHC2. It is as clear. Okay, so this will be represented to the T cell receptor. Suppose this is a T cell, this is a naive T cell. Okay, naive T cell means it's inactive T cell. T cell जब पहले आता है ना तो वो inactive ही रहता है, मतलब it's not immature. What you understand first, it is inactive. Immature T cell मतलब तो immature ही होता, तो it cannot get itself converted into TH1 or TH2. Okay, this is a T helper cell, naive T helper cell. Okay, naive cell or inactive cell. There is difference between inactive and immature cell. Immature cells cannot be transformed into TH1 and TH2 on its own. Okay, this is inactive cell which is waiting for some stimulation that can convert it into TH1 either or TH2. Okay, so this is the T cell receptor. Now again, I will not go into details of T cell receptor. You understand this is a toll-like receptor. Okay, and they basically recognize the molecular associated pattern. Okay, microorganism associated molecular pattern or damage associated molecular pattern. This is the T cell receptor. So as the antigenic peptide, the parts of the microorganism, parts of the antigen are represented on the crypt of MHC2 to the TCR, T cell receptor. It stimulates the TH cell. Okay, this is called the immunogenic synapse. Immunogenic synapse is the connection between the MHC2 represented along with APC and antigenic peptide with the T cell receptor and TH or T helper cells, or it can be other type of lymphocyte. Okay, it can be also TH1, it can be TH2, it can be cytotoxic T cells. Okay, so it is the total connection between this MHC and T cell receptor. MHC is situated on the antigen presenting cell, or T cell receptor situated on the T lymphocytes. Okay, this whole process is called the immunogenic synapse. Okay, so as it is I have previously told, and as it is an extracellular bacteria. Okay, extracellular bacteria, and the proteins are also exogenous, you can understand. Okay, it will be represented on MHC2. Okay, and it will be also represented to TH2 type. Okay, TH2, because in case of intracellular pathogen we induce TH1, but here we will induce TH2. Okay, TH2. This how this transformation will be taking place. Okay, first there will be, as this process is occurring, there is some extension on the antigen presenting cell. This is called the B7, and there is also another extension. This is called CD28. Okay, Now this B7 and CD28 will combine with each other. This is called the co-stimulatory signal that converts basically T cells into active, that will convert T cells into active Th2 cells. Okay. Now this there are more signals. Suppose if this antigen presenting cell is a macrophage, let it be macrophage, or any other antigen presenting cell. Okay, let it be macrophage. Suppose it is macrophage, so it will be releasing interleukin 1. Okay, interleukin 1. This interleukin 1 basically will act on the Th cells. Okay, and also help in its conversion. Okay, not only interleukin 1, there will be interleukin 4 from some unknown sources. We do not know the source. There will be interleukin 4 from some unknown sources that will also activate the Th cells. Now, what are the factors that is converting inactive T cells into Th2? One is interleukin 1, interleukin 4, interleukin 1 from APCs. Okay, interleukin 4 unknown source, it can be anything. Okay, then B7 and CD28, this is B7. You see B7 and CD28 interaction, B7 from the antigen presenting cell and CD28 from the T lymphocytes. Okay, and B7 and CD28 interaction also basically interaction between the antigenic peptide that is situated in the cleft of the MHC2 along with the T cell receptor. Okay, so these are the signals that is converting the inactive T cells into Th2 cells. Okay, I am explaining again. This is the APC, this is antigen presenting cell, there is extracellular pathogen, this let it be bacteria, and it is engulfed by the APC, and it is broken down by proteasomal machineries. There are smaller peptides, and these are representing on the cleft of the MHC2. Okay, MHC2. Why it is MHC2? Because it is extracellular pathogen. Okay, and the proteins are basically exogenous. It is not synthesized within the cell. Okay, if it would have been extracellular, intracellular pathogen means intracellular bacteria, but endogenous protein. Okay, sorry, exogenous protein. Then like tuberculosis and leprosy, we would have gone for MHC. What MHC? Okay, MHC2 only. So this is the whole process. Now, this antigenic peptide is represented on the T cell receptor. Okay, and this is the T cell which is getting activated into Th2 type. Okay, now this Th2 lymphocyte will produce basically interleukin 4 and interleukin 5. Interleukin 4 basically act on the B cells. Interleukin 4 is basically B cell growth factor. B cell growth factor. And interleukin 5 is B cell differentiation factor. Okay. So this B cell will get transformed into plasma cells, and this plasma cells will produce antibodies. This was Th2 type of response. Now what about Th1? Okay. The response of Th1 is basically delayed type of hypersensitivity. Delayed type of hypersensitivity. You can see from here in the Th1 cells that Th1 cells are basically responsible for intracellular pathogen like tuberculosis and leprosy and the exogenous proteins. Okay, exogenous proteins, because as T helper cells is connected to MHC2, so it has to be exogenous protein. Suppose this is an antigen presenting cell. Okay, can be anything, can be macrophage, can be dendritic cell, can be B cell. Now there is some TB or leprosy bacteria which is mostly intracellular, and the protein is broken down by proteasomal machinery, and it is represented on the cleft of MHC2. Okay, now this is represented on T cell receptor. Okay, T cell receptor, and this T cell receptor is situated on the head of basically T lymphocyte. Okay, now you see here, as it is intracellular pathogen, the signal will be for formation of Th1 cells. Okay, Th1 lymphocyte. The activated lymphocyte will be of Th1 type, not Th2. Okay, Th1 lymphocyte. Now there are also some other signals. Okay, like there will be production of interleukins. Okay, what are the types of interleukin? There will be production of interleukin 12. Okay, interleukin 12. The APC will produce interleukin 12. If interleukin 1, interleukin 4, okay, if interleukin 1, interleukin 4, and there is interaction, it will be getting converted into Th2 cells. Okay, but if the interaction is between interleukin 12 and Th cells, it will mostly get converted into Th1 type of lymphocytes. Okay, interleukin 1 also can be basically acting here, but the hallmark is interleukin 12. It is most important. Now these Th1 cells will release interferon gamma. This interferon gamma will again act on this macrophages. Okay, macrophages will engulf the antigens. Okay, and it will kill it by oxidative or non-oxidative mechanism. Okay, oxidative and non-oxidative mechanism. Okay, and there will be granuloma formation. This is the whole story of delayed type of hypersensitivity. Okay, today I am stopping here. Okay, thanks for the listening to my class. Thank you guys. Thank you बच्चों.